Plant kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggested the five kingdom classification, namely Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm and angiosperm under Plantae. Let us look at some classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed which were based on natural affinities among the organisms and considered not only the external features but also the internal features like ultrastructure, anatomy, embryology and phytochemistry. Such classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Numbers and codes are assigned to all the characters and the data is then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome numbers, structure, behavior and chemotaxonomy that uses the chemical constituent of the plant to resolve confusion, are also used by taxonomists these days. Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic and largely aquatic both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and wood. Some of them also occur in association with fungi, lichen and animals, example onslaught bear. For the form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Eulothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile female gamete and a smaller motile male gamete is termed oogamous, example volvox and fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy-rich compounds which form the basis of food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, that is, water-holding substances, example algin brown algae and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of the pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, and oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched form as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophyll. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic cell wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to the plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and a leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction is most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. The gametes are pyriform and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red alga because of the predominance of the red pigment, R-phycoerythrin, in their body. Majority of the red alga are marine with greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of the water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red alga are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar in structure to amylopectin and glycogen in structure. The red alga usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and is accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Rather table: Chlorophyceae, green algae, major pigments chlorophyll A and B, stored food starch, cell wall, cellulose, flagella 2 to 8 equal apical, habitat freshwater, brackish water, salt water. Phaeophyceae, brown alga, major pigments chlorophyll A and C and fucoxanthin, stored food mannitol, laminarin, cell wall, cellulose and algin, flagella to unequal lateral. Habitat rarely freshwater और वैसे mostly brackish water and salt water. Rhodophyceae, common name red alga. Major pigments chlorophyll A, D and phycoerythrin. Stored food Floridian starch. Cell wall cellulose, pectin and polysulfate esters. Flagella absent and habitat some are freshwater and mostly salt water. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called the amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in the plant succession on bare rocks or soil. The plant body of the bryophytes is more differentiated than that of alga. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true root, stem, and leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophyte are multicellular. The male sex organ is called the antheridium. They produce biflagellate antherozoids. The female sex organ, called archegonium, is flask-shaped and produce a single egg. The antherozoids are released into the water where they come in contact with the archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provides peat that have been long used as fuel and as packing material for transshipment of materials because of their capacity to hold water. Mosses, along with lichens, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rock, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophyte are divided into liverwort and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, barks of trees, and deep in the woods. The plant body of a liverwort is thalloid, example Marjantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. The asexual reproduction in liverwort takes place by fragmentation of thalli or by the formation of specialized structure called gemma. Gemma are green multicellular asexual buds which develop in small receptacle called gemma cups located on the thalli. The gemma become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on the different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, the spores are produced within the capsule. These spores germinate to form free-living gametophyte. Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs Antheridia and Archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of foot seta and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish in sandy soil conditions. You may recall that in bryophyte, the dominant phase in the life cycle is the gametophytic plant body. However, in the pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stems, and leaves. These organs possess well differentiated vascular tissues. The leaves in pteridophyta are small microphylla as in Selaginella or large macrophylla as in ferns. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobilae or cones, Selaginella and Equisetum. The sporangia produces spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of the specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophyte bear male and female sex organs called Antheridia and Archegonia respectively. Water is required for the transfer of antherozoids. The male gamete released from the Antheridia to the mouth of the Archegonium. Fusion of the male gamete with the egg present in the Archegonium result in the formation of the zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophytes, all the spores are of the similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and the microspores germinate and give rise to male and female gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygote into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in the evolution. Pteridophytes are further classified into four classes: Psilopsida, Psilotum, Lycopsida, Selaginella and Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Diopteris teres adiantum. Gymnosperms. The gymnosperms are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in others, cycas, small specialized root called corduroid root are associated with end-to-fixing cyanobacteria. The stems are unbranched as in cycas and branched as in pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperatures, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce the water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on the sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of the pollen grain takes place within the microsporangia. The cones bearing megasporophylls with ovules and megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree as in Pinus. However, in case of Cycas, male cones and female cones and megasporophyll are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. Nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on the megasporophylls, which may be clustered to form the female cones. The megasporophyll mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovule borne on the megasporophyll. The pollen tube carrying the male gametes grows towards the archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovule into the seeds. These seeds are not covered. Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms or flowering plants, the pollen grain and the ovules are developed in specialized structure called flowers. In angiosperms, the seed are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from the smallest wolffia to the tall trees of eucalyptus. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, and tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledon, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organ in a flower is stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divide by meiosis to produce microspores which mature into pollen grains. The female sex organ in the flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style, and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3 celled egg apparatus, 1 egg cell and 2 synergid, 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anthers are carried by winds or various other agencies to the stigma of the pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissue of the stigma and style and reach the ovule. The pollen tube enter the embryo sac where 2 male gametes are discharged. One of the male gamete fuses with the egg cell syngamy to form the zygote. On the other hand, the other gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. Because of the occurrence of two fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with one or two cotyledons, and the PEN develops into endosperm, which provide nourishment to the developing embryo. The synergids and the antipodals degenerate after fertilization. During these events, the ovule develop into seeds and the ovaries develop into fruits. Plant life cycles and alteration of the generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. Though these in turn divide by the mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alteration of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following pattern. Sporophytic generation is represented only by one-celled zygote. There is no free-living sporophyte. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically to form gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. An algae fucus species represent this pattern. In addition to all seed-bearing plants, that is gymnosperm and angiosperm, follow this pattern with some variation wherein the gametophytic phase is few to multi-celled. Bryophytes and pteridophytes interestingly exhibit an intermediate condition. Both phases are multicellular, however they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by the haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic/autotrophic independent but short-lived haploid gametophyte. Such pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus and algae is diplontic.